So our next speaker is Professor Sandhya Koushika from TIFR, and she's going to talk about regulations of axonal transport and conversations between biologist and a physicist.
Thank you very much for inviting me. I've been here for the past couple of days and it's been really nice, even though some of the talks I haven't understood, but the ones I have understood, I've really enjoyed myself. I'm a card-carrying biologist. There's no physics anywhere in my lineage. There's nothing quantitative ever anywhere in my life until I met Gautam. And I'm going to describe some of the work that we have done together somewhere in the middle of my talk. But I thought what I would do is give you a sense, a flavor of one aspect of what my lab works on. And over a decade, I have worked closely with Gautam, and I've also now started working a little bit with Sandeep Chaubey. And I have to say, as a biologist, I've learned an enormous amount. And that's why I've titled my talk as I have, Conversations Between a Biologist and a Physicist, and there need to be many more. I think the problems in biology are very complex, and I think to bring any meaningful understanding to them is going to require multiple approaches. All right. What I am is a neurobiologist. I study neurons, and neurons are cells that are present in your body, present in my body, which allows me to move my hand, to talk, to have percept when we eat something sweet and we like it. And the way the nervous system works is essentially through building a circuit. The circuit that it forms, and here's a spinal reflex arc, which any person would have studied sometime between 5th and 10th standard. So I assume at some point all of you have seen it. It's a reflex arc that we are all very familiar with, which we hit ourselves, either our edge of our elbow or our knee. There's a shooting pain and you withdraw your limb away from that stimuli. And that happens because you have neurons that sense the touch or that pain stimuli, which goes to a spinal cord and then goes down to have a reflex arc where your muscle withdraws. Now, supposing you are a sports person, and even though you're in pain, it's a goal right there that you want to shoot, you don't care, you go for it. So you have brain-mediated processes which can suppress some of these reflexes to continue to execute a certain motion. So what the nervous system allows you to do is sense information either internally, you're hungry, for instance, or externally, you're in pain, integrate that information and give you an output. Neurons are long, can be very long cells. Some of the longest cells start in your spinal cord and go to your big toe. Think of giraffes, think of elephants. They all have very long cells. And they are connected. The geometry is very much like an electrical wire. So what happens inside these wires? There's been a lot of interest to it through long periods of time, driven two strands of thought in neurobiology, one driven by disease. I think all of us live, including in India, in aging populations, and we look at things like dementia and Parkinson's and Alzheimer's. But we also look at injuries. In India, you have, as just yesterday, there was a discussion of road accidents, and a lot of those road accidents can lead to deficits in motor movement, deficits in what happens in the way we interact with the world because of damage to the nervous system. So that's one strand, often driven by war and our own aging populations. The other strand is to just understand the fundamental biology, right? So driven by both of those interests, Paul Weiss did a very, very interesting experiment a long time ago, and it's something worth thinking about still. It's a very simple experiment. He tied a little knot around a neuron, waited several weeks, and saw these bulges on either side of where he had tied the knot, showing that there was flow occurring in both directions. This is a neuron. This is the synthesis hub. This is where the communication hub is, where it'll talk to another cell, either the muscle or another neuron. And this is the wire that connects it. And you could see that there was flow taking place in both sides. A lot of interest in looking at this flow led to these sorts of experiments, which are very beautiful experiments, where you took the squid giant nerve, which actually Vijay talked about, squeeze out its contents, put it under a microscope, and what you saw were these roads, which are these cellular roads known as microtubules, along which various objects moved. And this is what we'll call cargo. And I'll talk about one kind of cargo today. What is the engine which allows these cargo to move along these cellular roads? Engine is called molecular motor. Here is a particular kind of molecular motor which works hand over hand, walking on the cellular road. Great. So we have the axon, which is the wire. Peel it apart, you have these roads. And along these roads, you have multiple types of cargo moving with the engine of the motor. So the entire process of moving things along the axon is called axonal transport. Very simple, like things moving along the road is called whatever traffic or transport that we see in our everyday lives. Essentially, for the purposes of the talk, we will focus on one kind of cargo, which is called the synaptic vesicle. Okay? That is the cargo which is filled with chemicals called neurotransmitters, which allows us to execute these complex motor functions or any sort of function associated with the nervous system. Okay? So I want to be as far as possible jargon-free if I can make it. What you do in this factory or in this cell, in this part, is actually make these little compartments which have the right composition on them. You'll hear a little bit more about it. Recruit the engine, which is the motor. Get it out along the axon onto the cellular road. Move along it, maybe fall off, hop onto the same road, hop onto the end of the other, hop onto another road, come to the communication hub, which is the synapse, dock there, release chemicals. And then you start the whole process back again and going back. I won't look at that today. So the very simple question, if you're interested in any aspect of this problem, if you talk to a general public, would say, are there traffic rules in neurons? And the three pieces of the puzzle that I introduced was the cargo, that is what is moving, the environment, which is the axon, which has all of those cellular roads, which are microtubules as an array, and the motors, which is the engine which moves things. So the first aspect of my work, of my lab's work, dealt with looking at the cargo, specifically looking at synaptic vesicles. Why are these they're obviously very important. Without synaptic vesicles, you will not be able to breathe. And if you don't breathe, you don't live, right? Because your neuromuscular junctions over here, your neuron muscle junctions of your diaphragm have to move up and down. Many people, like people who have ALS, die not because they can't walk around, but because they can't move their diaphragm muscles. Okay. The synaptic vesicle is a very unique and unusual organelle in some ways, I like to think, in that we know a lot of information about it, but what's very defined, and you can forget about all the rest of it, saying that there are many, many proteins which are present on it, many different kinds of fats or lipids which are present on it, but the numbers of each of those molecules is very well understood and invariant. That is actually pretty remarkable when it comes to biological systems of these nature. We won't get into that today, but what me as a lab decided to do is to try to understand what makes synaptic vesicles, and using an approach called genetics, using genetic screens, we identified two different steps which are exceptionally important in forming the synaptic vesicle. So the kind of experiments, and you will see a lot of movies today in my talk that we do, is to mark various kinds of proteins inside the cell and then image them. So here we have marked a marker for synaptic vesicles, as well as some other thing which is actually called the Golgi, but it doesn't matter very much. And what you'll see in normal healthy neurons, you'll see mostly green. This is synaptic vesicles coming out of the cell body and getting on those microtubule arrays, cellular roads. What you see in this particular mutant is both of them are mixed up and moving along. So what we deduce from this and many other experiments, that this is the first step where you ensure that some of these proteins don't get out into the axon in these carriers. That's the first step. And that step is actually retaining Golgi into the cell body and keeping it at the Golgi. And by the way, Golgi got the Nobel Prize for his staining technique, and there were other issues associated with it. If you ever feel like, do read his Nobel lecture. Fantastic work, but also a lot of bitterness because he ended up being wrong. Step 2. The step 2 is the next step after you get out of the Golgi. There are some other proteins which are present, like lysosomes. And what you see generally in wild type is you see only synaptic vesicle proteins coming out in the axon. Lysosomes are stuck here. And in this particular mutant, we see that they're actually moving together. That's why they look yellow, because the red and green are present together. So we had a hierarchical series of steps to begin to get a defined organelle which is present in the synapse and filled with neurotransmitters. What relevance does this have to movement or axonal transport? And here is this very simple experiment that we did, and I'll sort of walk you through it. This is the step 1 defect, and this is a step 2 defect. This is a synapse where synaptic vesicle proteins are present. And this is how it's present along the axon, present in the cell body. What happens when we remove the synaptic vesicle motor or the engine? Here's what we see. In a normal animal, you remove the synaptic vesicle motor, there's nothing, no synaptic vesicles that come up to the synapse. Fantastic. This is what you'd expect. You can't breathe, you can't move, you will eventually die. What happens when you have a step 1 defect? These are not proper synaptic vesicles, but you can still bring some aberrant stuff all the way to the synapse. What happens when you have a step 2 defect? You can't bring it all the way to the synapse, but you can bring it some way into the axon. What you deduce from that is that you can have different types of composition of vesicles or cargo that comes out, and the number of the right kind of motors that probably get onto it is critical in how far that cargo goes. And that's the only message I want you to think about, that cargo matters, and cargo matters because of the types of motors that get onto it. So in a normal animal, in a normal vesicle, in a healthy animal, you will have a lot of the synaptic vesicle motor present on it, which is the engine. In this particular step 1 mutant, you get a lot of different kinds of motors present on it. What we think in the step 2 mutant is that you have fewer motors of the right kind, the engines present on it, and potentially few of the other kind. All right, so we'll come to the heart of my talk. We'll start looking at the environment. You've done the first job of making your cargo and coating with the right kind of motor. What about the environment? The other way to ask the question is, are there constraints on cargo flux in an axon? And the answer is absolutely, you have to consider it, because even the extremely old EM pictures, and these are some of my favorites from someone who used to be the leader in the field, Nobutaka Hirokawa, and you can see how crowded the environment in which the organelles are moving. So here's a mitochondria, here's probably a synaptic vesicles, you have large cargo, you have small cargo. These, by the way, are the motors and the engines that move these different cargo. If you think that this kind of meshwork underlying it is the only issue that you see, the other issues that you see is look at this nerve cord. Each of these axons is a different diameter. So then organelle, or here when you look at this neuron, which is C. elegans, which is the organism I study, you see one neuron with a lot of different tracks and another with very few and with a lot of open space. And so cytoskeletally or inside the neuron, in my view, an axon is like an Indian road. There's a lot of heterogeneity and a lot of mess that occurs. Structured, sensible mess, but still a mess, where you can have changes in diameter of the axon, changes in number of microtubules, or the number of roads that are available, and other kinds of cytoskeletal elements here shown as pectin rings. But there are many others which are not mentioned here. So in this complex environment, I already showed you one movement of how cargo moves in neurons. What happens if we look at cargo movement, specifically synaptic vesicles? And this is the critter I work on, C. elegans. You see cargo moves in both directions, and some cargo doesn't move at all. I actually started showing Gautam some of our data, wanting to understand a different question, which we still haven't made much headway in. But he pointed out that a lot of the cargo were not moving. And it was really useful conversation when he insisted on trying to understand that, because we really hadn't taken a systematic approach in the field to try to understand where stationary cargo came from. So after establishing it was not an artifact, we went on to characterize it like all good biologists. So one of the things that we did was to try to see where these stationary cargo were present. And what we saw is there would be places where there would be stationary cargo which wouldn't move, they would disperse, and then you'd have something come and stop at the same location. So think of it as a bend in the road that we drive normally, or a place which is very close to a marketplace and you'll always get crowding and things would stop at that region. So it made sense for us to try to see whether they were present in certain internal structures. And one of the structures that we looked at was something called actin, which is present like a meshwork in neurons and also present as rings. So we don't really know what the geometry of this actin is. All we can say is this cytoskeletal element is enriched over there. And what we saw is that a lot of the stationary cargo, and ignore all of these other things, just focus on this bar, you see that a lot of the stationary cargo are present at actin-rich regions. The rest of it is for completeness, for an experiment to see if various cargo behave the same way, various neurons behave the same way, and neurons from different animals behave the same way. And the answer was yes. And here was a piece of very nice analysis that my then student Parul did. It was her idea, where she went on to say, since we know that cargo stall where there's actin or some kind of cytoskeletal crowding feature, let's ask how moving cargo behave in this location. So where you did not have any actin enrichment, most of the cargo will move through that region without any problem. Think of it as highway traffic. When you have only an actin enrichment, what you saw was some amount of the cargo did not do so well and tended to stop. It was not quite as moving as highway traffic. And this was striking when she looked at places which had just a stalled cargo. This would be the equivalent of a truck which is stopped in front of you on a road, and another truck comes behind. A far larger number of the cargo stopped. And finally, the worst traffic block that you got was when you had both actin and a stalled cargo. That is, you had this tangled cytoskeletal feature as well as a synaptic vesicle. What this suggested to us was that this kind of stalling in the axon could perhaps arise due to just physical crowding. You do not need to invoke motor-based mechanisms. And in addition, what we saw is a similar aspect that we saw in another class of neurons in another animal altogether. So we think that physical crowding in the neuron might be critical for stalling. So here is a movie where I can show you actin-rich regions, and here's a vesicle that comes and stops over there. So you can sort of get a flavor of what you're actually looking at and what we are counting. So cargo stop at crowded regions. Great. That's not, you heard Mithun yesterday for those of you who were there, and he was talking about physical crowding along DNA, there with protein complexes. Here you're looking at a completely different case because vesicles are, several nanometers in diameter, 50 to 70 nanometers, and some large organelles can go up to a micron in length. So you're talking about a different size scale, but conceptually, it's crowding. So is it? And we're talking about crowding in healthy neurons. There's been a line of thought, as I said, about thinking about disease, where many investigators have thought that traffic congestions and jams are relevant in sick neurons. And the reason they have thought that is because of these variety of experiments looking at human postmortem samples, mouse models, or you can even C. elegans, which is what we use, models of various diseases. And one feature that you will see is these swellings with lots of proteins and organelles filled in. So the idea very simply was that you will now stall cargo transport. And we thought naively that there are two possible ways in which, because what people had seen was a reduction in cargo transport as the disease progressed. So the ideas were that changes in transport can either be causal, correlative, or a consequence of many of these diseases. So the simple hypothesis that occurred to us was increase in stalled cargo caused by increased in physical crowding. Because the clear observation was as the disease progressed, there was much more stalled cargo. And the second is you might, in healthy neurons, have mechanisms to bypass these traffic jams and congestions, and these are compromised in axonal transport in diseased neurons, right? So do we give you an answer? We give you a way to think about it. We don't have an answer yet. So the other way to think about it is, how do you get past traffic congestions in healthy neurons? And here's where Gautam and his student Reshma, as well as Vinod's work came in. So they sort of benchmarked the data that we had through these simulations, and what they showed was the following. If you have multiple microtubules and a cargo doesn't do anything, everything will stop. All cargo transport will stop unless when they come to a traffic jam, they can take some sort of decision. Either change the lane like we would, or do a reversal and sample that space later. So what I call is bypassing traffic jams requires local decisions in space where you can decide whether you want to change lane and get onto another microtubule track, for instance. And here is the data which comes from what happens when you reduce the rate of sidestepping. You actually find that you have an effect on how much current occurs. And here's a better graph where you now look at reversals, where the way I think about reversals is you have a stalling taking place, a vesicle comes there, it reverses, and it can sample the same space after a period of time where something might have changed locally, allowing it to go past that particular traffic jam. So you can have relaxation in space or relaxation in time. So what you see with cargo transport, this is regular, there's no reversals, your net current drops to 0 if you do not allow for any reversals. And therefore, the number of stalled cargo increase. How does it, so it's very hard in an experiment to look at state changes where you look at a cargo changing tracks. But you can experimentally measure reversals. And here is a vesicle that's reversing. And you can sort of see this is an actin-rich region. It has to get past it. This vesicle goes back and forth, and finally it'll get past and come out. So you can do these sorts of measurements and count it. This is how you would convert this kind of movie into a kymograph track where you do reversals. And what we see is that we get reversals at where stationary cargo present or away from, so not very different from each other. So does it matter where you reverse in your simulations? So this is what a biologist call, is your mechanism of reversal triggered at a particular location, and what is the effect of triggering? That is, you trigger a reversal mechanism when you see a crowded location. And in fact, human drivers, in the absence of Google Maps, make local decisions by looking at what's right there in front of them, right, when they drive on Indian roads. That's not what vesicles in the neuron are doing, okay? Keep that aside. But you can ask that question in the simulation, thankfully. And what you see is if most of your reversals take place where you have a crowded location, you have the maximum amount of current taking place. If they take place more or less like an experiment, anywhere along the neuron, you don't have that bias, you have a moderate amount of current. And the least amount of current is if you do it completely opposite, that is, you actually do your reversals away from places which are crowded. And that's a very interesting thought. Maybe different cargo use different strategies. We need to investigate that. We don't have an answer. So now let's circle back to the disease condition. And because we have a disease model in C. elegans, we can look at that. We can look at reversals rates. We can look at number of cargo that are stalled, and we can look at net current. When you look at reversal rate, what you see from, a preteen or an adolescent to an adult going on to a middle-aged adult, your reversal rate goes up and continues fairly high in normal animals. In your sick animals, your reversal rate goes up a bit but begins to decline. In conjunction with that, I'm not saying that there is a direct one-to-one role for this, because we do read to many more experiments, but it's very suggestive that your number of stationary cargo slowly increase in this animal, and the net current begins to go down in keeping with the reduction in reversals. And in fact, you can say further and say your reduction in reversals seems to occur earlier than the kinds of changes that you see in current. So from this work, we concluded that stationary cargo are a feature of axonal transport, and traffic congestions occur in healthy neurons, much like they would occur in even normal roads, and reversals and potentially its locations might matter. The last piece of the puzzle is motors, and I'll be done in a couple of minutes. The motors are the engines which move synaptic vesicles, and I thought I would come down to it because it's the last piece of the rules of traffic that you have to consider. What we know with the synaptic vesicle motor is that it gets activated, it moves and carries cargo to the end of the neuron, and what we had shown earlier is that it gets degraded. And we had this long series of experiments trying to find proteins that regulated this process, and specifically I will tell you what we were looking for regulation. We had developed this assay with Kaustubh, who was an engineer when I was at NCBS, where we could pick up that there was an anterograde bias in transport, where if you injured the neuron, you saw that the motor actually accumulated when it was coming from the cell body, but very little motor came back. So we wanted to understand what went on behind this biased anterograde transport. And what we found by doing, again, genetic screens was a gene that was very, very important for controlling how many motors went in what direction. And that's clearly going to be very important because we already know that the types of motors on the cargo surface are very important, and that depends on the nature of cargo. And here what I'm trying to say is the number of motors might actually matter. And in fact, looking at this particular mutant, okay, let's see if I can play this as well. This is your wild type. You will see a few puncta moving here, which are bright. What you will see in your mutant animal is many, many, many more very bright puncta of motors, which are presumably on the cargo surface. And we can convert that into these kind of distance time chemographs and quantitate roughly how bright these puncta are. And here's where this whole project we discussed greatly with Amitabh Nandi, who's in IIT Bombay, a colleague of Mithun's, and Debashish Chowdhury in Institute of Physics, and I think would have never dared start this collaboration except for the long history that I had with Gautam, which gave me the language to speak to physicists. And what we saw in these mutants, so they developed a model to explain the flow of motor, and what they did were essentially fits to our intensity data. And what was striking, and I'm not showing a lot of the data that went into this kind of experiment and the kind of model that we did, that they developed, but what was very clear from their fits was that they couldn't get this fit in the mutant condition unless they added a cooperativity term. And I'll come to that back in the last slide. But what was very clear from this kind of from the movies as well as this kind of analysis, that cargo seemed to have very many more motors on their surface. So what would you expect would happen when you had very many more motors on the cargo surface? What you expect is that the motor will walk further before it falls off the microtubule, simply because you have many, many more engines. And the answer, indeed, that is exactly what we saw. What we suspect is there are fewer motors normally. When you get rid of this particular modification in this particular gene, we think there are many more motors on the cargo surface as supported by our imaging with the motors. And what we see is the cargo now begins to walk much, much further. Okay. So coming to the end of my talk, what we realized from looking at the simulation was that cooperative binding may actually play a critical role in active cargo transport. I know that sometimes when you look at fits, you people wonder what is the relevance of that fit for biology. But for the first time, for our group, we have come to the thought that when you have multiple cargoes present on the surface, especially in this case, multiple motors present on the surface, what we now think is there's some kind of partitioning that happens which allow them to come together in some way through interactions with each other. What they are is something that is going to drive our experiments. And here's an example of how the theory opened the door for us as an experimentalist to consider new kinds of experiments. So what are there traffic rules in neurons? The answer is yes. The two most important pieces of the traffic rules is what is being moved, which is the cargo itself. Because what is being moved decides how it's being moved. Because what is being moved decides what motors you put on the surface. And the number of motors you put on the surface decide how far you go and how you go. What does the environment do? Immensely important. But in normal healthy neurons, you have a crowded environment. So the role of environment for a healthy neuron is uncovered much more in disease states. When you have disease states, you no longer have the ability to deal with your normal crowded environment. So I think the major rules are the cargo and the motor and the environment if you're not doing too well. And none of this work would have been possible without long-term interactions with Gautam's group and his office in IMSA and the wonderful coffee that used to be here, Reshma and Vinod, and more recently Vidur, who's my student, who's essentially driven most of that interaction. The interaction with Gautam has also inspired a lot of people in my lab to do more quantitative work, which has been very nice, and I thank you for your attention.
Thank you very much, Professor Koushika. So we have a small memento for you.